We're now going to move on to the third speaker in the session, who's Dr. William Harris, who's president of the Fatty Acid Research Institute in Sioux Falls. And you can now share the screen, William.
Thank you. Assume it's visible and if people can see this. Appreciate being part of this presentation. I've learned a lot so far, and hopefully this will add a little bit more to the surround sound of this topic we're having today on nutrition, or lipid nutrition, and COVID. So just to press on, let's see if I can get this to advance. That would be helpful. Just to prove that there actually are falls in Sioux Falls, I want to show a picture of the falls of the Great Sioux River. This is pretty good. It's not Niagara, but it's for the northern plains, it's not bad. So let me begin with a very 35,000 foot look at this question. This is a nice little paper that was published this year from a group that actually did some in silico work trying to predict the effects of omega-3 fatty acids on the spike protein itself. But in the introduction to their paper, they summarized very crudely, but very interestingly, I think, data from the intake of omega-3 fatty acids around the world and worldwide fatality. And they looked at, again, the number of countries in each of these 6 regions are shown over here on the left. And I'm getting my pointer working here. Here we go. And here on the right is a very crude graph. We're not doing statistics here. This is just a sense that here's the rate of regional fatality from COVID-19 from WHO data. And then across the axis here is the average intake of EPA and DHA in each region. It obviously doesn't prove that there's a relationship, but it's interesting to see that there may be an ecological system or signal around the world that there is a role to play for omega-3 fatty acids. And of course, my topic is to talk about potential mechanisms of action of omega-3. And I always like to be sure that there is an action to have a potential mechanism for before we even bother looking at potential actions. But obviously what we've seen from Philip and Charlie so far, there's very strong evidence for potential. So let's look at some of the evidence that there actually is an effect. Overview, I'll look quickly at some of the associations between omega-3 fatty acids and inflammatory biomarkers. Again, repeat something of that Philip said about the effects of omega-3 on inflammatory biomarker levels. I'll give the results of a pilot study that we published in January looking at blood levels of omega-3 as a predictor of risk for death from COVID. And then refer back to some data that Philip, I think, also referred to. I'll present it in a slightly different way, looking at the randomized trials with omega-3 fatty acids in acute respiratory distress syndrome settings. A year from now, there will be 10 meta-analyses on omega-3 and COVID, but for now, they really aren't. So we have to use surrogate lung-based diseases to look at this. Mechanisms of action, we'll probably go through this very quickly because it's sort of been covered already with Charlie's presentation, as well as Phil's setting it up for many of these these molecules that have tremendous in vivo action across wide spectrums of systems. And I will show actually the same slide Philip showed on membrane effects. It's always good to see these things 2 or 3 times so you might actually remember them. And that's the way we'll end up the talk. So I want to introduce, first of all, a metric that we've been using for some years called the omega-3 index, because it will come up again in a moment. The omega-3 index is a blood marker of omega-3 status. It's the amount of EPA and DHA in red blood cell membranes, and it's expressed as a % of total fatty acids. So in this cutout of a red blood cell, if we look at the lipid bilayer, in this cartoon, there are 4, the red phospholipids are containing omega-3 fatty acids. So there's 4 out of 64 in this particular cartoon. That ratio is simply 6.25%. So the omega-3 index would be 6.25% for this, which is a little higher than what you see in most Western countries and quite a bit lower than what you see in Japan, et cetera. The desirable omega-3 levels, we think, or we proposed some 15 years ago, be under 4% is where you definitely don't want to be. Over 8%, perhaps 8 to 12% is a good place to be based on a variety of data sets. Typical for the US and Europe, somewhere between 4 and 8, roughly 5 or 6 is probably an average for those regions. So that's the omega-3 index. Again, a target of 8% would be optimal. Under 4% is not so good. And with that introduction, let's look at some data we published some years ago with the Framingham cohort. We looked at the correlation between the omega-3 index and multiple inflammatory biomarkers. And this was in fundamentally healthy people drawn from the community there in Framingham, Massachusetts. 2,700 people, and we looked at age and sex adjusted and multivariable adjusted relationships between the omega-3 index and these 10 circulating biomarkers of inflammation, which are, obviously this is not all of them, one could look at, but it's quite an array that spans a variety of different systems. And in each case, if we look here in the multivariable adjusted correlation, the correlations are not strong, they're not high, but these are also healthy people, not surprisingly, but they're all statistically significant and they're all inverse, meaning the higher the omega-3 levels, the lower the levels of these pro-inflammatory markers. So there's some suggestion that simply having higher omega-3 reduces levels of inflammatory markers across the board. Philip also showed something like this, excuse me, but data showing that if you actually give EPA and DHA, you do lower inflammatory cytokines. And this paper was 35 patients with leg ulcers who were given 2.5 grams of EPA and DHA a week versus a placebo for 8 weeks. And simply looking at IL-6, IL-1 beta, and TNF-alpha in each case, the omega-3s group saw a decrease in the inflammatory markers. The control group did not, and the difference in change was statistically significant. And the authors concluded that long-term EPA-DHA therapy may be a safe, relatively low-cost dietary intervention to prevent or assuage inflammaging, a relatively new term. Obviously, the inflammation associated with aging and the diseases that greatly diminish the quality of life. So again, there is a randomized trial data that omega-3s will lower these markers. I mentioned a study we published just recently. This was done in collaboration with Dr. Arash Asher at Cedars-Sinai in Los Angeles. We were able to obtain blood samples from about 100 individuals. You know, Philip's presenting data on 450,000. We're doing 100 here. So you got to look at all sides of this. 100 patients who were admitted to the hospital with COVID-19. We were able to get samples of that blood and measure the omega-3 index in those people. And we then divided the groups into quartiles, 25 people each, and looked at the risk for death. And so here in this category, I'd really just like to focus on the bottom area, which is where the most important information was, I think. If we compare individuals who had an omega-3 index under 5.7%, which was the lower 3 quartiles. So this was a low omega-3 group in general. Compared to quartile 4, which is the highest 25% in the group, which is over 5.7%. The relative risk for death in hospital was 0.25, not quite statistically significant, 0.07. But good enough to suggest that there may be something to having a high omega-3 level upon entry to the hospital. This says nothing about the effects of potential treatment with omega-3 once a disease has been contracted. But it does imply, as Philip was talking about, a preventive model where higher omega-3 may reduce risk for adverse outcomes. Getting back again also, I think, to some data that Philip was referring to, a meta-analysis of omega-3 in acute respiratory distress syndrome trials. So these are RCTs looking, and this is, I think, part of Philip's Cochrane review. which I need to look at in more detail. I had not seen it. I had only seen this one, but in this is making the same point, I think, in these tables. Here we're looking at a beneficial effect of giving omega-3, and this is the outcome is what's called the PaO2 FiO2 ratio, which fundamentally is the arterial oxygen level versus the inspired oxygen intake. So somewhat the efficacy of getting oxygen into the blood from the lungs. And in these trials, there was a significant beneficial effect of that metric at 3 to 4 days of treatment, also at 7 to 8 days of treatment. This is early and this is late. And finally, looking here at the effects on the need for mechanical ventilation, significantly reduced risk for that. So again, making the point that here in a different lung disease, inflammatory lung disease, there is an effect, randomized trials of omega-3. So mechanisms of action. This is a slide we've been using for many years, published by Daria Mozaffarian and Jason Wu 10 years ago now. And this was again published in Journal of the American College of Cardiology, and it was a great review of what was known about mechanisms in those days. What we see here is that there are, when you look at body system wide, reduced risk of arrhythmias, increased myocardial oxygen uptake, reduced systemic vascular, and endothelial dysfunction, so improved function of the endothelial surface, and then reduced risk of thrombosis. And these, I think, are mechanisms that still play a role or being shown to be effective here in the inflammation setting as well. Also in that paper was this nice drawing of again, mechanisms at the cellular level. And again, 10 years ago, reduced inflammation because of increased resolution, as Charlie just described, from protectins, resolvins, morasins were not known at the time. And there was a reduction in inflammation as well. The omega-3s were believed to change membrane fluidity and thus affect raft activity, which affected the actions of multiple transmembrane proteins and ion channels, fatty acid transporters. Down here is a proposal that the not proposal, but the note that the omega-3s do reduce risk for inflammatory eicosanoids made from arachidonic acid. And then if the omega-3s can affect actual transcription of important nuclear factors that will produce the inflammatory cytokines. And so we've seen this. This was, of course, known 10 years ago, and now it's known in much more detail. To just summarize again, as Philip has already done, that there are a tremendous number of molecules made from the LOX, COX, and cytochrome P450 systems, not to mention the endocannabinoid systems that produce generally pro-resolution, anti-inflammatory markers from EPA and DHA. So these obviously can play a role in reducing disease. I think we've become, and it's certainly become more clear recently that one of the major mechanisms by EPA and DHA is by blocking the activation of NF-kappa B, which has been called the gatekeeper of the inflammatory response. And to the extent that that is true, they would certainly have beneficial effects throughout the entire body, as this is the primary pathway in. And here again is the slide Philip showed, just to reiterate again that the presence of higher levels of EPA and DHA will reduce the amount of arachidonic acid that is in the membrane, reducing the substrate for production of this in the 2 series and 4 series, 2 series cyclooxygenase metabolites and 4 series leukotrienes and a variety of P450 metabolites. EPA and DHA can, again, produce resolvins, protectins, morasins, which resolve inflammations. They can alter raft assembly. The particular sections of the membrane that are responsible for sensing inflammatory stimuli and reducing the transmission of that signal. And again, by reducing NF-kappa B, will stop a tremendous variety of cytokines and chemokines from being synthesized in the first place. So the mechanisms of action are quite clear, and it's pretty obvious the way that this particular group of nutrients can play a role in this disease. The slide I showed at the beginning was the international comparisons of death rates in omega-3 intake, and that was an introduction to this paper. And this is the primary point of this paper by Vivar Sierra and colleagues, was to suggest a novel mechanism, novel to me anyway, and thought I would share it with you, although this is certainly by no means proof. But this was done, this was an in silico, not an in vivo, not an in vitro, but an in silico experiment, much the way drug manufacturers design new drugs. They look at molecular models on computers and look at the potential interactions of drugs with receptors and how they may bind well or not. In this particular paper, they were looking at the hypothesis that the presence of DHA in the plasma may actually interact with the spike protein itself. In this way, they show here on the left that if you have a low EPA and DHA intake, the spike protein interacts with the ACE inhibitor. It has to be in an open or unlocked configuration in order to interact with the ACE2 receptor and then be brought into the cell. Once brought into the cell, this entire cascade of inflammatory action occurs. What they showed in their, again, in silico analysis was that if DHA, they're focused particularly on DHA, if it's present, DHA can be bound in the active pocket of the spike protein, and it will lock it in a closed formation, a conformation. And if the spike protein is in a closed conformation, it will not be picked up by the ACE inhibitor, excuse me, ACE receptor, potentially preventing the actual import of the viral particle into the cell. This, of course, needs certainly more work to document that it's true, but the in silico experiment suggested that this may be a mechanism, one of many mechanisms by which the omega-3s may reduce the overall adverse effects of the disease in humans. So let me wrap up quickly. I think that we talk about potential protective mechanisms. It's clear anti-inflammatory and pro-inflammation resolving properties of omega-3 are clearly at work in playing a role in reducing the adverse effects of COVID. There are also, as others have mentioned, antithrombotic effects. We didn't go over those, but EPA and DHA back from the actually in 1978, when Dyerberg and Bang proposed that EPA would compete with arachidonic acid for the production of thromboxane, is one of the earliest presumed effects, known effects of omega-3 fatty acids. So to the extent that antithrombosis is helpful in COVID, it certainly could be playing a role this way. Omega-3s do improve vascular health, vascular responsiveness. They can increase the transmission of oxygen, efficiency of myocardial use of oxygen. As I mentioned briefly, they are antiarrhythmic in many settings, at least in the ventricular arrhythmias. What's happening with atrial arrhythmias is another question. And then this highly speculative but fascinating idea that maybe the omega-3s can essentially block the uptake of the virus by maintaining the spike protein in a closed conformation. So those are a kind of an overview of the possible mechanisms, and I think we're ready for questions.
Thank you, William. That's another beautiful presentation there, and we have bang on time, so we have lots of time to ask some questions.
Great.
I can't see any on the board at the moment, but I'll just start the ball rolling again. Yeah, it's interesting to see, although you commented you had a relatively small number of patients from that study at Cedar-Sinai. Nevertheless, it's appeared to be a very positive effect. Yeah, as somebody from the vitamin D field, we see this quite a lot in our own studies, but it's often, how can I put it, tempered by the idea that somehow the disease itself has impacted on whatever you're measuring. In this case, the sort of omega index. Do you think that's possible? Is there a mechanism by which that could occur, where you have an acute inflammatory disease is driving the omega index down?
It's pretty hard to imagine I mean, you can imagine a situation where somebody is feeling ill for quite some time. Their diet might change. Their use of supplements might drop off if they were taking them. Just not feeling well could in the long, but the turnover, the half-lives of omega-3 fatty acids in membranes is much longer than the time frame to get infected and get sick with COVID. So in that sense, I don't see a physiologic or biochemical in vivo effect. It's hard to imagine.
And well, on a similar note, if you look at other sort of acute care settings, have you seen similar observations there with people with, I don't know, severe burns or whatever, faring better if they have a better omega index?
Crohn's has not been looked at, but in general, I mean, we published a paper last week looking at probably the most important clinical outcome, which would be death. Death from any cause, and we did find that a higher omega-3 index across a group of 45,000 people, 17 different trials or observational trials. There was a significant drop off in the higher the omega-3 level, the lower the risk for death, and applied not only cardiovascular disease, but it applied somewhat to cancer as a cause of death. And interestingly, that we had 3 categories of death besides total mortality: cardiovascular, cancer, and then everything else, sort of the kitchen sink of everything else people die from. And a higher omega-3 was very clearly related to a lower risk of death from everything. So it suggests a system-wide benefit of higher omega-3 levels.
Yeah, indeed. I don't know if we have any. I'm searching around for questions on the board. Can't see any at the minute, so I've got time for one quick question. Which I mean, the sort of in silico model of the prevention of coronavirus 2 uptake is really exciting. But is this something that looks as if it could be quite specific? I mean, has anybody looked at other viruses which obviously have a different topography than the coronavirus? So what do you think this could be something that could be a universal antiviral thing? Or
yeah, it's hard to say. I mean, I'm getting way out of my league on this level of biochemistry. But the authors of this paper said that they were driven to look at this question because a previous study had reported that linoleic acid in silico was sort of behaving the same way, that it was able to keep this the spike protein in a closed conformation. Okay. So they decided to look at the omega-3s. But yeah, there's a lot more to look at in this world.
Clearly, yes, as you pointed out. So we do have usual things right at the end. We have a question come through from Anne Menier, who said, as many was already known or at least to suspect on the potent role of W3 in the regulation of inflammation. Why does it take so long to be tested on COVID? Not quite sure I understand the question there. Excuse this naive question. As much, I guess, was already known, or at least suspected on the potential role of W3 in the regulation of inflammation. I guess why is it taking so long for this to be incorporated into COVID-19 studies?
That's so long. It's all relative, isn't it?
It's 1 year.
Fast, at one level. I think, well, Adam's going to talk next about, at least last I saw, something like 14 trials ongoing in this world, and they should come to fruition quite quickly, I would think. The endpoint here is now we're not looking at 10 years of death, we're looking at 3 weeks. So I think we'll have a lot coming. I don't know why it took longer to get data versus vitamin D, although vitamin D has been measured in many more people for much longer, I think. So we'll just have to be patient, and it'll come.
Okay, on that note, I think we will then move on to the next speaker. But thanks again, William. That was an excellent presentation.